Good morning, everyone.
Good morning.
How are you today? Hope you had a nice weekend. Okay, well, welcome back, everyone, and I hope you're safe and sound and in a warm environment. It's very chilly these past several days, isn't it?
Yeah.
Okay, some exciting news. If you haven't heard, HAC has made the decision that starting next week, Monday, January 31st, folks, we are going back to school. We are going back to school, in-person instruction on the Lancaster campus next Monday, okay? So keep our fingers crossed that things go well. But this is pretty exciting because we haven't really had classes on campus for nearly 2 years, okay? Because of this darn COVID situation. So that's really exciting. And so please spread the word among your classmates, that are in class, that once again, we'll be back on campus next Monday. So next Monday morning, I'll be saying hi to you in person, okay? That's wonderful news. Okay. So we're going to get started in earnest taking notes today out of chapter 1. Are there any questions you would like to ask before I get started? Okay, gang, here we go. Of course, we have lecture today, and this morning is also our first lab together. So we're really going to start moving here. Okay, I'm going to share my screen with you. All right, and so typically, at least this week, what you'll see is a note-taking document up on the screen. I will try to keep up some video on the right-hand side of the screen and a chat window. So if you want to communicate with me, feel free to do so. Okay, so, our first time together last week was all about reviewing the syllabus and class expectations, what's going to happen. Today, we're going to get started in chapter 1. So let's begin. We got to begin somewhere. Let's start with some opening definitions. You're taking a course that's called Anatomy and Physiology. So I'll define anatomy and physiology for you. By the way, I want to encourage you to have your digital textbooks open at this time because I might refer to pictures and figure legends in the digital textbook. And so it will be helpful if you have that open today. Next week, when we get back to campus, please bring in your computers and tablets and things so you can access your digital textbook during our lecture time. Okay. All right, so excuse me for a moment while I check the chat window. Okay, so here we go, folks. Anatomy. Anatomy is the science of body structures. What is that bone? Well, that bone is the femur. What is that muscle? Well, that muscle is the rectus femoris. What is that organ? What is that ligament? What is that tendon? What is that blood vessel? What is that nerve ending? What is that ganglion, right? The anatomy of body structures. Anatomy is the science of body structures and the relationship among or in between those body structures. Okay. Physiology doesn't focus on the body structure as much as it focuses on how the structures function. What is the physiology? How do the molecules interact? How do the cells interact? What is the chemistry driving the ability of the body to do what it does? So physiology is the science of body function. Okay, physiology attempts to answer the question, how does the body work? How does it do what it does? Okay. All right, so anatomy is the science of body structure. We're going to be tackling that primarily in lab. Physiology is the science of body function. We're going to be tackling that primarily in lecture. Okay, so there's two major topics in chapter 1 that I want to cover with you. We probably won't finish it all today, but we're going to get started. The first major topic is the levels of structural organization within your body. And so I would turn your attention to figure 1.1, figure 1.1 in your textbook. Okay, levels of structural organization in the human body, figure 1.1. And we need to think about this a little bit. Okay, so, if I were to tell you, that a baseball is smaller than a car, you would be like, well, of course it is, right? Because you've lived the number of years you've lived, and you have that everyday life experience. You have the intuition to know on our everyday scale of interacting with the world, the relative size of one object compared to another object, right? An airplane is larger than a car. A baseball is smaller than a car, right? A spoon is smaller than a gallon of milk, right? We all know these sorts of relationships, and it's kind of even silly to talk about them. However, when we start talking about really, really tiny objects that are inside our bodies and make our bodies possible, that our bodies can function the way they do, typically I find that students lack the intuition they need to understand how your body is organized. And so that's what figure 1.1 is trying to describe to you. Rather than simply listing out figure 1.1 in your notes, what I'm going to do is go to an alternative website and show you the relative size of things from very, very tiny objects to much larger objects as a way to help you begin to develop your intuition of how objects are related from a size perspective, okay? So I'm going to use the Genetic Science Learning Center website, which is at the University of Utah. And I'm going to put a hyperlink in your note-taking document. Here it is. You can see it on your screen at this time. Let me get this into your notes. Okay, so it's hyperlinked and ready to go for you when you study on your own. So you can see at this time, you can see the Genetic Science Learning Center webpage. It has several major themes: genetics, evolution, human health, cell biology, plants, neuroscience, ecology, science tools. So you might have some fun playing around with this on your own private study time. Again, I have provided the web link in the notes that I'm taking and will give you later today. I'm going to go to the cell biology section, the portion that says amazing cells. I'm going to go into amazing cells. And you can see when you click into that tab, there's a whole bunch of content here. You can explore this during your own study time. I am going to go to the topic along the left column called cell size and scale. Cell size and scale. Okay. And what we're going to do is just take a little tour, and we're going to start at very, very small things. Very, very small things. Okay. So again, we're trying to get you comfortable with how the body is organized. Well, it's all going to start with atoms. It's all going to start with atoms. And so, when we talk about atoms, we're talking about all that stuff on the periodic table of elements. And we'll do more discussion of that in chapter 2 in about a week's time. But for right now, we'll start with carbon atoms, okay? So I'm just answering a question in the chat window. Tristan, the notes will be provided on Wiley Plus. When you go into Wiley Plus, the lecture portion of Wiley Plus, go to the Files tab and you will find today's notes. By the end of the day, I will post them for you. Okay, so our bodies are basically made of molecules which themselves are made of carbon atoms. So we start with the carbon atom as the smallest unit of matter which we commonly find in our bodies. Carbon atoms are 340 ppm. Okay, ppm stands for a picometer. Okay, 340 picometers in diameter, 340 picometers wide. So let me ask you this. From using the information that's right in front of you, please tell me in the chat window, how long is 1 picometer? How long is 1 picometer? Take a look in the bottom right corner of the web page, folks. How long is 1 picometer? Okay, so we're starting to talk about things that are very tiny. And so we get some numbers that are actually very small numbers, okay? All right, so I'm just going to pause a moment. Thank you, Percy, for contributing. Veronica Cutter, fantastic. Okay, thanks, Muhammad, right? So 1 picometer is 1 trillionth of a meter. So let me ask you this. Let me ask you this in the chat window, coming next. How many inches are in 1 meter? How many inches are in 1 meter? Okay, it's probably been a while since anybody's asked you that, right? Because at least in the United States, we don't frequently talk about meters, do we? Okay, so meter is a unit of length, part of the metric system, more formally part of the System International Units, and that's what we use in science. So how many inches are in 1 meter? Okay, any guesses? Now, if you're at home and you're Googling it, that's okay. I don't mind that. I just want you to participate, okay? All right. So yes, for those that are still looking it up, a meter is about 39 inches. 1 meter equals 39 inches. Okay. So what I want you to think about is, imagine for a second you have a yardstick between your hands. Okay, so here we have a yardstick between my hands. A yardstick is 36 inches. 1 yard equals 36 inches. Add 3 more inches to it, and now you have a meter stick. So if you were to take that meter stick and you were to cut it up into 1 trillion evenly divided segments, and you counted up 340 of those tiny little segments, 340 of those tiny little segments would be the distance across a carbon atom. Carbon atoms make up many of the molecules in our bodies. Okay, so atom is basically the smallest unit of matter that we will talk about in our course. That's not to ignore the fact that you know about things like electrons and protons and neutrons, which are even smaller than atoms. Okay, so we have carbon atoms, and we're starting to get just a little bit bigger, okay? So here we have molecules. We have methionine, adenine, here's glucose and some things. We're getting to the point where we have molecules. So I will point out something. Let me ask this in the chat window. Here we go. Which is larger, a carbon atom or a water molecule? Which is larger? Okay. Take a look at that. Okay. take a look at the dimensions. According to the dimensions, isn't the carbon atom larger? The carbon atom is 340 picometers wide. The water molecule is 275 picometers wide. Okay, so, generally speaking, we would expect you to appreciate that atoms are smaller than molecules because molecules are made of groups of atoms. That is generally true 99.99% of the time. We can sometimes have an exception. For example, the water molecule is made of two small hydrogen atoms, an oxygen atom, and those atoms are so tightly squeezed together due to the forces of attraction between the atoms, the chemical bonds, that the overall molecule actually has a diameter that's smaller than an individual carbon atom, which is a giant whale-sized kind of atom, okay? So just be aware of that there is an occasional exception to the rule that molecules are larger than atoms. Here you see the perfect exception. A water molecule can in fact be smaller than some of the larger atoms. All right, so we have carbon atoms. Atoms are tiny. Then we progressively get to larger structures called molecules. Methionine is a molecule. It's in fact an amino acid. So let me ask you this. Here we go. What type of organic macromolecule will contain methionine? Methionine would be a part of something larger. What's that larger thing that I'm fishing for? So gang, if you don't know, it's okay. I don't expect that you do know, but right now I'm trying to determine, what your background really is, which is why I'm fishing with different questions. Okay, so does anybody have an idea what type of macromolecule, larger molecule, will contain methionine? Okay, so take a look. If you don't know, take a look at Cutter's response in the chat window. It's proteins, and he is absolutely right. Methionine is a part of proteins, okay? Because methionine is a type of amino acid. If you don't know that, it's okay. You will know it by the end of chapters 2 and 3. Adenine. Adenine is another type of molecule. Okay, so let me ask you this. Here we go. Try this one. What type of organic macromolecule will contain adenine? Where do we find adenine inside cells? As part of what larger thing? Okay. Any guesses out there? Okay, so if you haven't heard of this, adenine is a part of nucleic acid. Nucleic acid. Thank you, Percy, for your contributions. You are right, Percy, that you are going to find this inside the nucleus. So adenine is part of nucleic acid, and the two major types of nucleic acid inside cells are DNA and RNA. DNA and RNA. Okay. All right, so folks, we're just taking a tour right now. I don't expect you to know the answer to all my questions. It's okay. So we start with carbon atom, which is super small, and then we link carbon atoms and other atoms together to build larger molecules, like methionine, like adenine. Let's continue our tour here. Here we have glucose. Glucose, is C6H12O6. It's got a diameter of 900 picometers. Okay, and you know that glucose is commonly used to manufacture biochemical energy inside cells. So let me ask, just to see what your background is, I'm going to ask a question in the chat window. What is the name of the process that breaks down glucose to make ATP? Do you have any background on that? Do you know about that? What is the name of the process that breaks down glucose to make ATP? Okay, Cutter, I appreciate your participation. You are not wrong. I'm just looking for something a bit more precise. So catabolism is the breakdown of something. Sarah, mitochondria are relevant because mitochondria are the powerhouse organelles of the cell. So Sarah, that was a good answer, just not quite what I'm fishing for. Tristan, homeostasis is a nice idea. We're going to be talking about that a little bit later. Still not quite what I'm fishing for. All right, so folks, the answer to my question is glycolysis. Glycolysis. Glycolysis and excuse me for a second while I spell that right. Okay, glycolysis is the breakdown of glucose. You'll learn more about that in chapter 3. Okay, so we started with carbon atoms, very small. We're getting progressively larger with groups of atoms we call molecules. So far, we've talked about 4 molecules: a water molecule, methionine, adenine, and glucose. Okay, I'm going to start moving a little bit faster, but you get the idea that we have atoms, and then we put groups of atoms together to build molecules. All right, so next, we have phospholipids, which are part of the cell membrane's building blocks, okay? The cell membrane, the plasma membrane of cells made of phospholipids. We also have things such as antibodies here, okay? Antibodies have been in the public conversation in the news because of our COVID pandemic, right? Everybody wants to know if they have antibodies against Omicron or any of the COVID variants. Okay, so antibodies are certainly going to be part of our immune system, part of our body's response to protect ourselves. Antibodies are actually specialized immune system proteins. Antibodies are specialized immune system proteins, okay? You can see here that an antibody is roughly 12 nanometers in length, roughly 12 nanometers in diameter, okay? So let me ask this in the chat window. How long is 1 nanometer? How long is 1 nanometer? Okay, all you have to do is look at the bottom of your web page, and it's right there for you. Okay. All right, it is. In fact, thanks everybody. Sarah, Kenneth, Cutter, Ashton, Veronica, good job. It is 1 billionth of a meter. So a nanometer is larger than a picometer. A nanometer is larger than a picometer, okay? So an antibody is 12 nanometers long. We have tRNA here. tRNA is 7 nanometers long. tRNA is short for transfer RNA. Transfer RNA brings amino acids to the ribosome to manufacture proteins. If, as you sit at home, you're thinking to yourself, he's talking Greek, I don't know any of these words. That's normal. It's normal, okay? By the end of chapter 3, you will know these things, okay? All right, let's talk about hemoglobin here. Hemoglobin, 6.5 nanometers long. Hemoglobin is the compound in your red blood cells that delivers oxygen and sometimes carbon dioxide to various locations of your body. Hemoglobin is a gas transport compound found in red blood cells. Okay, here's a question for you. coming in the chat window. Here we go. On average, how many copies of hemoglobin are found inside a single red blood cell? What would you say? Hemoglobin is 6.5 nanometers long, so it's pretty tiny. And hemoglobin fits inside cells like red blood cells. So how many, on average, how many copies of hemoglobin are found inside a single red blood cell? Do you have any idea? Give me a ballpark range. Give me a ballpark range. Okay, thank you, Sarah, for participating. Any other guesses? All right. Thanks, Percy and Veronica. Appreciate it. Folks, let me type the answer into the chat window. Okay. Here it comes. 280 million copies of hemoglobin in one red blood cell. So, folks, that you guessed, you were a little on the low side. Okay, you're a little on the low side. Okay, but that's why we need to talk about these things, right? That's why we need to do that. Okay. So hemoglobin you'll be encountering when we talk about blood, and frankly, you'll talk about blood more in AMP2 than you will in AMP1. That's a next semester topic, okay? All right, we're getting a little bit bigger, a little bit bigger, a little bit bigger. Okay, next we have a ribosome on the screen, which is 30 nanometers wide. You can see a ribosome is larger than an antibody. It's larger than transfer RNA. It's larger than hemoglobin. Okay, so let me ask you this in the chat window. What material do ribosomes manufacture? What material do ribosomes manufacture? What do they make, gang? Okay, thanks, Cutter. Anybody else want to take a guess? Okay, thanks, Tristan. So we have a disagreement here, don't we? All right. So thanks, everybody. Ribosomes, in fact, manufacture protein. That's what they do. Ribosomes manufacture protein. So that antibody you see on your screen, which is a special protein, was made by a ribosome. The antibody was made by a ribosome. Okay. All right. So, again, we're just starting the conversation to get you used to thinking about very small things with a foreign vocabulary that you're not used to thinking about, but you will be used to thinking about it, in 4 weeks when you're getting ready for your first lecture exam. Okay, let's get bigger. Hepatitis virus, 45 nanometers long. This virus is larger than a ribosome, which is larger than an antibody. Hepatitis virus, is one, there's different forms of it that often causes liver damage. We have rhinovirus, which is 30 nanometers long. Rhinovirus is one of many viruses that create cold symptoms in human beings. I'm going to get a little bit larger now. A little bit larger. Okay, here we have an influenza virus, right? The flu virus, 130 nanometers in diameter on average. HIV, also 130 nanometers in diameter. Human immunodeficiency virus, the virus that creates AIDS, acquired immunodeficiency syndrome, in folks that are unfortunate enough to be infected. Okay. Here you have a thing called a coated vesicle. Coated vesicles are things that enter the cell. They enter the cell and the cell builds a coat around it. On average, these are about 90 nanometers in diameter. Up here in the upper left corner, we have a phage, which is the same thing as a bacterial virus. It's about 70 by 200 nanometers in diameter. Let me bring it on screen so you can fully see it. There it is. Okay. So folks, that's something to realize, that even bacteria get infected with viruses. Even bacteria get infected with viruses. Okay. If we're talking about a cell that's on our planet Earth, it gets infected with viruses. Okay, all cells get infected with viruses. Let's continue. Okay, we have a measles virus, 220 nanometers long. All right, here we have some other objects starting to come into view here. We have a mitochondrion and an E. coli bacteria. You may know that mitochondria are the sort of the power plants of the cell. This is where the bulk of our ATP is manufactured. Okay, Percy, I see your question. So Percy is asking, what happens when a bacteria gets infected with a virus? That's a very complicated question, Percy. And so let me just give you two ideas. Typically, when any cell, including a bacterial cell, gets infected with a virus, the virus will change the expression of the cell's genes. So the cell's physiology will change because the cell is now turning on and turning off different combinations of genes because the virus is present. Let me give you one specific example. So Percy, are you aware? You're aware of the disease that people get called strep throat. Okay, strep throat is caused by a bacterium called Streptococcus pyogenes. If a person has strep throat and the bacterium Streptococcus pyogenes is itself infected with a certain virus that is known to exist, then Streptococcus pyogenes, when it's infected with the virus, can, instead of causing strep throat, it can cause scarlet fever. Okay, so even the different, if you have a bacterium that is or is not infected with a virus, you can create different diseases in a host animal. Okay. All right. So on the screen again, we have mitochondrion and we have E. coli bacterium. Okay, E. coli, is present in your large intestine. It helps you maintain a healthy internal environment in your body. So let me ask you this in the chat window. Coming in the chat window. Do you notice that mitochondria and E. coli bacteria are very similar in size? Do you see that? Mitochondrion is 4 by 0.8 micrometers. E. coli is 3 by 0.6 micrometers. Do you see that? They're very, very close in size. And so when you look at that, when scientists saw that, they started to think to themselves, I wonder what that's all about. And long story short, it turns out, folks, that scientists believe that mitochondria inside your human cells is really primitive bacteria. Mitochondria are symbiotic bacteria that live inside our cells. Mitochondria are symbiotic bacteria that live inside our cells. How did they get there? Well, you'd have to go back millions of years in evolution to figure that out. So nobody truly knows. But there is fairly strong, compelling evidence to believe that mitochondria are of bacterial origin. Okay. So Percy is asking, so when the term good bacteria, that's what we are talking about. Generally speaking, yes, Percy, E. coli would be considered a good bacterium when it's located in the large intestine. Yes. Okay, so notice that we've changed from nanometers when we were talking about hemoglobin and viruses to micrometers. So we started out with the carbon atom, 340 picometers. Then we went to transfer RNA, 7 nanometers. Now we're at E. coli bacterium, 3 micrometers. A micrometer is 1/1,000,000th of a meter. So a micrometer is larger than a nanometer. A nanometer is larger than a picometer. Okay? Am I going to beat you up with the math of this on an exam? No, I'm not. I'm not going to beat you up with the math, but I am going to beat you up with your intuition. Do you appreciate that a virus is larger than an amino acid? Do you appreciate that an amino acid like methionine is larger than a carbon atom? Do you appreciate that a mitochondrion is larger than a measles virus? Do you appreciate these things? That I will beat you up about, okay? All right, so let's continue. We're going to get a little larger here. By the way, there's a lysosome thing on the screen. A lysosome is a tiny little bubble structure inside your cells which recycles worn out material. So if you have a damaged molecule that's no longer useful, the cell will put it into the lysosome and the lysosome will recycle it. Okay, let's get bigger. Let's get bigger. Okay, here we have some interesting things. We have an X chromosome, which we know if you're a lady, you have 2 copies of an X chromosome in every one of your cells. They're about 7 micrometers long. We have baker's yeast, which, you know, maybe you or your mom or grandma use baker's yeast to make bread at the house, which is very tasty, right? So that's 3 by 4 micrometers in terms of length by width. The red blood cell on the screen, 8 micrometers wide. You remember that question I gave a few minutes ago? How many copies of hemoglobin fit in a red blood cell? The answer was 280 million. So 280 million hemoglobin molecules fit in a red blood cell that has an average diameter of 8 micrometers. Okay. Folks, if you become a teacher someday, the best way to scare your students is start to talk about math. Okay. So that's the way we do this. All right. Very good. So relax. I don't care if you know the math. I care if you get the intuitive understanding of what things are smaller or larger than other things. That's what I care about. Okay. So let's get bigger. Let's get bigger, bigger, bigger. Here we have some interesting things. We have a sperm cell here, right? A sperm cell which is 60 micrometers long. That would come from the male of our species, right? We have a skin cell here. So on the surface of your arm, your face, your scalp, you have cells that are on average about 30 micrometers in diameter. Here we have a photoreceptor on the right side. Photoreceptor is very thin. It's very thin, 2.5 micrometer width, but it's very long, 100 micrometers. Okay, so let me ask you a question in the chat window. Here we go. What part of your eye anatomy contains photoreceptors? What part of your eye anatomy contains photoreceptors? Anybody know? Okay, good job, Percy and Tristan. Absolutely. Okay, thanks everybody for contributing. If you take a look, the majority consensus answer is in fact retina, and that's correct. Photoreceptors are found as part of the retina. The retina is a layer of nervous system tissue that lines the wall of the eye, the inner wall of the eye. Okay, let's get bigger. Okay. Next, we have a human egg cell, right? A human egg cell is gigantic Okay, this big circle, this big blue marble up here is the human egg cell, 130 micrometers wide. Look how big the egg cell compares to the sperm cell. The egg cell is giant compared to the sperm cell. All right, we have a paramecium, which you might have played with in middle school growing up, 210 by 60 micrometers. Going to get a little bit bigger now. Amoeba proteus is a type of protozoa, 500 micrometers in diameter on average. If you've ever gone swimming in a lake or pond in the summertime, you know, wherever you've been, undoubtedly these microorganisms have been in that lake and pond water, paramecium or protea, paramecium and amoeba proteus, they've been there. So they were on your skin, they were covering your eyeballs, they were in your mouth. If you've ever swum, you know, if you ever went swimming in one of these bodies of water. Okay. All right, let's continue, get bigger, bigger, bigger. All right, now we're getting some things you can start to really appreciate, right? A grain of salt. A grain of salt that you have in your salt shaker on the kitchen table. The average grain of salt is going to have a diameter of about 0.5 millimeters. A millimeter is 1,000th of a meter. So a millimeter is larger than a nanometer. A nanometer is larger than a millimeter is larger than a micrometer. A micrometer is larger than a nanometer. A nanometer is larger than a picometer. Let's get bigger, 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 bigger. Here we have a sesame seed you might have on a bagel. Here we have a grain of rice, which you might cook for dinner tonight, 8 by 2.5 millimeters. Here you have a coffee bean, 12 by 8 millimeters if you enjoy coffee. And then your typical font, 12-point font on a word processing document you're writing up for your English teacher. Well, that's how big it's going to look. Okay. So again, folks, don't worry about the numbers. I'm not going to beat you up about the numbers, but very much do worry with understanding the relationship between atoms, molecules, and things that are larger than molecules, okay? All right, so we're going to document some of these things in your notes at this time. I'm going to put some of these ideas in your notes. Right now, I was showing it to you. Now I'm going to type it into the class notes, okay? So I need to change my screen. Do you have any questions right now? So let's do this. We're back on the notes, and I'm going to do this. I'm going to organize your notes so that things are going from small to large, okay? So first thing we have are atoms. Examples of atoms include carbon atoms, hydrogen atoms, oxygen, etc., etc. Basically, every atom on the periodic table of elements. Next, if you take atoms and you group them together, you build larger structures such as molecules. Okay, so in the website that I just showed you, we had many larger molecules. We had methionine, which was an amino acid. We had adenine, which is a nucleotide. Okay, so folks, in the chat window, tell me the name of any other molecule you know of. Tell me the name of any molecule you know of. Tell me the name of any molecule you know of. Anything at all. Okay, so Veronica, thank you. A ribosome is not a single molecule. It is not a single molecule. It is made of many tens of molecules that are grouped together like Lego bricks. So I won't put a ribosome here. but that's a nice answer we can use later, Veronica. Okay, let's get some more ideas. Give me some other. Okay. So Jessica, okay. Jessica, you said nitrogen. So that's complicated, Jessica. Nitrogen is an element on the periodic table. So if it's an individual atom, it's a nitrogen atom. If nitrogen combines with two or more nitrogen atoms, then we form a nitrogen gas. So I could put down nitrogen gas as a molecule. Okay. Some of other molecules, glucose, that's a good one. Okay, good. DNA is a molecule. Good. Calcium oxide, good. Okay, table salt. Good, very good, folks. Okay, chlorophyll, Sarah, that's a giant molecule. Good. You're getting the idea. Nice. Remember, what we're trying to get you to do is develop an intuition of the size of atoms compared to the size of molecules. All right, good job. Let's keep going here. Next, if we take molecules together, we can build a layer of structure that's not really talked about in figure 1.1 of your textbook, but I'm going to talk about it here in your notes. And I'm going to refer to this as nanostructures and nanomachines. By nanostructures and nanomachines, I'm talking about objects that are smaller than cells, but larger than molecules. So nanostructures and nanomachines would be things that are made of groups of molecules, okay? And examples here would be things like the lysosome. Lysosome would be a nanostructure, okay? That was in the website that I showed you. Let me see if I can find it again. It was... There it is, right? 1 micrometer, that's the lysosome. That would be an example of a nanostructure, okay? Another example of a nanostructure, or really a nanomachine, is the term that Veronica, you wanted to use, which is a ribosome. A ribosome, which is the protein manufacturing hub of a cell, is a nanomachine. Nanostructures and nanomachines are larger than individual molecules because they are made of multiple groups of molecules that interact. Okay, so that's nanostructures and nanomachines. Next, we have cells. Okay, here's where I think you start to get a little bit more comfortable, right? So in our website that we shared with you, we had red blood cells. We also had individual bacteria like E. coli bacteria, right? By the way, probably because it's useful, let me hop back here. Nanostructures and nanomachines. Nanostructures and nanomachines would include viruses. Nanostructures and nanomachines would include viruses, okay? All right, so back to cells. Cells like red blood cells, E. coli bacteria. Now, folks, what I'd like you to do is tell me the name of any type of cell that you are aware of. Give me a cell type, a cell name, any kind of cell that you know of. Type that into the chat window. Okay, so we have white blood cells for our immune system. Veronica, a leukocyte is a white blood cell, so thank you for that. Epithelial cells, you bet. Nerve cells, you bet. You're getting the idea. Now remember, okay, Courtney, muscle cells, good. Remember, in our website, we also had the following cells. We also had paramecium and amoeba proteus. Paramecium and amoeba proteus. So let's put that in there. Okay. All right. Good job, everybody. Good job. It's a nice list to get started thinking about these things. All right, so we start with atoms, then we group them together to build molecules. We group molecules together to build nanostructures and nanomachines. We group the nanostructures and nanomachines together to build cells. And then we take the cells and we group them together into multiple cells at one time to build tissues, right? Tissues. And here, there's lots of tissues in the body. We're going to do a tissue chapter, chapter 4 in the future. Right now, however, I'll just define the 4 major types of tissues in the body. There's 4 major categories. They are epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Those are the 4 major types of tissues in the body: epithelial, connective, muscle, and nervous. There are many subtypes within each of those categories, and we'll talk about them more deeply in chapter 4 of the book. So you take cells and you group them together to build something larger, which is a tissue. If you take the tissues and you group them together, you will form organs. And here I find students really do develop more comfort, because you might not used to be talking about tissues, but I bet you know things like your heart, your liver, your gallbladder, your spleen, your brain, right? These are organs, right? So think of any major organ you are aware of, and that would be correct, okay? So heart, liver, kidneys, gallbladder, etc., etc. By the way, folks, simply because I know it's coming up, in about a half an hour, we're going to be in lab together, and we're going to start introducing you to the skeleton. Skeleton, of course, is made of many bones. Please appreciate that every one bone of your skeleton is an organ. Every bone of your skeleton is an organ, okay? Individual bones of the skeleton are organs. Okay, if we take our organs and we group them together into something that has common functionality and purpose, then we would build organ systems. And as you start to look in your digital textbook, notice table 1.2. Notice table 1.2 in your textbook, and you will see that table 1.2 highlights a variety of organ systems. There are 11 organ systems that make up the human body, and I'm not going to list them all out for you, but you can see them for yourself. So we have things like the endocrine system. We have things like the digestive system, right? You have the nervous system, etc., etc. You are going to learn many of the systems in AMP1, and you're going to learn the rest of them in AMP2. Okay, so, we're just getting this journey started, and about 8 months from now, give or take a little bit, about 8 months from now, you will have finished this traumatic experience. Okay, very good. So let's continue. If we take organ systems and we put them together in a unified way, we will actually complete the entire human body, the organismal level of figure 1.1, and I'm just going to say the complete human body. Okay. All right. Okay, gang. So, please spend a little bit of time thinking about these things during your private study time. You want to start getting familiar with atoms, molecules, nanostructures, nanomachines, cells, tissues, organs, organ systems. Start to understand the relatedness of these objects. Do you have any questions at this time? Okay, so there's a lot of content in every chapter of your textbook. And, I just spent 56 minutes talking about one figure. Okay. And so what I'm trying to tell you is there's no way that I can lecture on literally every topic in every chapter. That would require a month to cover one chapter, and we just can't do that. So typically I will give you 1 week on each chapter, and I will pick out some selected topics of each chapter to lecture on. Please understand that if I don't lecture on something that's written in your textbook, I do still expect that you read it, okay? And it is still fair game for an exam, okay? So even though I'm not listing out all 11 organ systems, you certainly want to read them and start getting familiar with them, okay? And truth be told, you're going to get some homework starting this week through your lab work that's going to start to emphasize some of the information in chapter 1 of your textbook. Okay, so we're going to give you lots of support and lots of challenges to learn this content. Okay, so the second and final topic of chapter 1 that I will spend time on is the idea of homeostasis. So homeostasis. So let me ask you a question in the chat window. Here it comes. What is the section number of homeostasis in chapter 1? What's the section number? Okay. I just want to be sure that you're seeing it, okay? So take a look in your digital textbook. Do you see that homeostasis is section 1.4, 1.4, okay? So we're going to spend the rest of our time today talking about homeostasis. And I'm confident we won't get it done today, but that's okay. We'll finish it next time, okay? So, okay. Homeostasis, section 1.4 in your textbook. Let's talk about this. So let's start with the definition, okay? Homeostasis refers to the state of balance, or you might prefer the word equilibrium. The state of balance or equilibrium inside the body that is maintained by regulatory mechanisms that work at different levels of your organization. By that, I mean cells, tissues, organs, and organ systems all work together as a team to maintain homeostasis. Okay, so homeostasis refers to the state of balance or equilibrium, if you prefer that, inside the body that is maintained by regulatory mechanisms working inside cells. Well, I'm not typing too good here today. Inside cells, tissues, organs, and organ systems. Okay, so let me read that again. Homeostasis refers to the state of balance or equilibrium inside the body that is maintained by regulatory mechanisms working inside cells, tissues, organs, and organ systems. Okay, so that's just a starting point. Let's give you an example here. For example, consider the body condition of resting blood glucose. Resting blood glucose. Okay. Resting blood glucose is a body condition that should be maintained in a state of equilibrium. It should be maintained in a homeostatically healthy way. All right. So I'm going to ask this in the chat window. Okay. What is resting blood glucose supposed to be in healthy persons? What is resting blood glucose supposed to be in healthy persons? So let me contextualize this question. When you go to sleep tonight and you sleep 7 hours and you wake up before you have breakfast, what should be the glucose concentration in your bloodstream? That is the resting blood glucose level. When you wake up tomorrow morning before you have breakfast, if we were to measure your blood sugar, what would it be? Okay. All right. So I'm starting to get some numbers in the chat window, and I very much appreciate that. Anybody else want to contribute? Okay, so we're seeing some numbers that are reasonable, and that's wonderful. Let me say, and I'll pick on you because that's what I need to do, right? I need to point out some weaknesses in your knowledge, okay? Everybody wrote down some numbers, and Percy, you gave me 1 unit, but that unit is incomplete. That unit is incomplete, okay? All right, so what are the correct units to express blood glucose? What are the correct units to express blood glucose? Okay. Percy, you could do that. You could do milligrams per milliliters. If you're seeing the chat window, Percy is saying milligrams per milliliters. Percy, there's no reason you couldn't do that, but typically it's not expressed as milligrams per milliliters. Tristan, you are correct. It's typically expressed as milligrams per deciliter. Okay, so resting blood glucose should be, depending on the textbook you use, you might get slightly different values stated by the experts. So don't worry about if your values are a little bit different than mine, but roughly 70 to 110 milligrams of glucose per deciliter of blood plasma. Okay, milligrams per deciliter of blood plasma. Now, okay, so Percy, you're asking what is the comparison, right? A deciliter is 1/10 of a liter, and a milliliter is 1,000th of a liter. Okay, so I want to show you something in your own textbook. I want to show you something in your own textbook. Folks, in your digital textbook, I want you to locate Appendix C at the back of the textbook. Locate Appendix C as in Charlie. Locate Appendix C as in Charlie in the back of your textbook. And I'm going to ask you to read a question for me. I'm going to ask you to read information off the chart. Okay. So let's do, let me just pick one here. So here we go. Using the information in Appendix C, please tell me what, I'm typing this into the chat window. Okay, so give me the proper numbers and the proper units. Here we go. What is the normal creatinine level for females? Normal level for creatinine in females. Creatine. What is the normal level? Okay, so we'll just give you a few moments to see if you can find that. Tristan, you did a good job. Okay. All right. So just be sure that you can read that. Okay. Good. Good, 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 good. Okay. All right. Good. So remember, folks, if you give me a number without a unit, you're going to lose credit. If you give me a number without a unit, you're going to lose credit because the units absolutely matter, right? You can't tell me 4. What is 4? Is it grams of sugar? Is it bananas? What is it, right? The numbers have got to have a unit so that it makes sense. Good job. Okay, any problems locating Appendix C? It should be at the back of your textbook. I don't expect you to memorize these things. The reason we have books and computers and internet is so that you can find it, okay? All right, so, okay. All right, so Jessica, I see your comment that you're struggling to find it. Jessica, all the appendices are at the rear of the textbook after chapter 29. So if you get to chapter 29 and you go beyond chapter 29, you should start seeing the appendices, okay? All right, any other questions? Okay, Tristan, thank you for saying so. So apparently some versions of the book could have slightly different numbering to the appendices. Okay. Okay. Thanks everybody, and I appreciate you helping each other. That's what I want you to do. That's fantastic. All right, so let's continue our conversation about homeostasis. So I'm going back to chapter 1 here. I'm back in section 1.4 in my book. That's page 9. Okay. So we need to talk about the control of homeostasis. All right. So there's a lot of ideas here that need to be communicated. First off, homeostasis of any body condition, such as resting blood glucose, is always monitored and adjusted. It is always maintained by something known formally as a feedback system. Okay, so you have the idea of feedback systems on pages 10 and 11 of your book. Certainly figure 1.3 would be relevant here. Okay, so control of homeostasis is achieved through feedback systems. So let's define a feedback system. Okay, a feedback system refers to all the components of your body, and those components, that word component is very general. That component could be a cell, could be a tissue, an organ system that work together as a team. So all the components of your body that work together as a team to monitor a body condition, such as resting blood glucose levels, and which report information to a control center, okay, which then determines whether an adjustment in the condition is necessary. Okay, so there's a lot of wordiness here, which is why we need to define it, then we need to give you examples. All right, so let me define this perhaps a little bit more cleanly. A feedback system refers to all the components of the human body, and again, cells through organ systems that work together as a team to monitor a body condition, determine if a body condition is out of homeostasis, and if necessary, take corrective action to return the body condition to homeostasis. Okay, once again, a feedback system in general refers to all the components of the body, cells through organ systems, that work together as a team to monitor a body condition, determine if a body condition is out of homeostasis, and if and when necessary, take corrective action to return the body to homeostasis. Okay, so what we'll do next is talk about the components of a feedback system. Okay. All right. So first we have the receptor. The receptor. A receptor is a body structure which almost always contains a nerve ending. And if you think for a moment, it's going to contain a nerve ending because we really want our nervous system to be aware of what's happening inside the body, right? Your nervous system is the master command and control center of the body, so it needs to be aware of what's happening in the body. So a receptor is a body structure which usually contains a nerve ending that monitors changes in a body condition. And again, for body condition, I'm talking about blood sugar, blood pressure, pH levels, etc., etc., and which communicates information about that body condition to a control center. Okay. A receptor is a body structure that monitors changes in a body condition and communicates information about that body condition to a control center. Okay, so an example here. Okay, an example here could be a taste bud. A taste bud would be a receptor. A taste bud is a receptor. Okay, we kind of ignore it and don't give it much thought on a daily basis, not really, but our tongue is loaded up with these receptors called taste buds. And taste buds are made of clusters of cells in which in the middle of that cluster there are neurons. So when you put food in your mouth, the molecules that make up the food, whether it be sugar for sweetness or salt, for saltiness, or other molecules that might give bitter sensations or whatever it might be that we're experiencing, these molecules will bind to the cells of the taste bud, activate electrical signals along the neuron that's part of the taste bud. That electrical signal goes up to the brain and you say to yourself, oh, I just put something bitter in my mouth. I don't like that, so I'm going to spit it out, right? I'm going to spit it out. All right, so the receptor is the taste bud. Okay, in terms of blood sugar, well, what's the receptor? Well, that's complicated, but we could make a strong argument for your pancreas organ being a receptor, because the pancreas is the organ that releases insulin, right? So, the idea of a receptor can actually be fairly complicated, and we'll just keep it simple to get started with. All right, so let's see here. All right, it looks to me like the time is 9:15, so I suppose I really should stop. But folks, do you have any questions so far about homeostasis, feedback systems, or the idea of a receptor? We're not done with this conversation, but I'd like to answer any questions you might have at the moment.
Professor, I want to know what's going on with one of the girls I bartend with. She got COVID and now she really doesn't have smell and taste. What's happening in the body that her receptors are not functioning like her taste buds?
Yeah, that's an excellent question, Veronica. I don't know that I could truly answer that, or if anybody could truly answer that. There is some evidence to suggest that the COVID virus will infect cells of your nervous system. So even if your taste buds are working correctly and they're detecting the molecules of the food that you eat, it's very likely and possible that the brain cells that receive the information, because of the presence of the virus, the brain cells are no longer working correctly, so you cannot consciously perceive the flavors of food that you're ingesting, okay? So the blunt answer is nobody probably truly knows, and kind of the somewhat of an answer is the virus is influencing the nervous system. Okay. One more question in the chat window, then we'll take a break. From Percy, are stem cells the cell name before differentiation takes place, or stem cells the name of a cell that can undergo differentiation changes? Okay, all right. So good question, complicated question, because Percy, there are different stages of stem cells, but I would say this, at the most basic primitive level, stem cells are the kind of cells you have before differentiation takes place. Okay.
Okay, thank you. Yeah, in the textbook, when it just basically writes it. I read it a couple times, and the way I read it, I couldn't tell if, because it said this is called at the end, like after it went through the process. So the way it was written, I didn't know if it was saying after the cell changes, it's now called a stem cell, or the cell that could change is the stem cell. So it's the cell that can change is the stem cell.
Correct.
Okay.
Yes.
All right, thank you.
Sure. All right, gang, it's 9:17. And so we need to break at this time. So we have lab at 9:30, but I like to try to honor the 15-minute window of break here. So my clock just went to 9:18. So folks, we will start lab at 9:33. We will start lab at 9:33. So take yourself a 15-minute break. This Zoom channel will stay active, but I'm going to mute my audio and video. 9:33, I'll come back online with you, okay? So take a break. See you in 15 minutes.